Hello, professors. Professor Tai is here, and we are gathering in the conference and get together to attend this conference. And Professor Tai will address the remark in Chinese.
So every professor is online. Can you hear us? Yes, I can hear. I will start.
Hello, everyone. I would like to welcome everyone to tonight's discussion, and today, or good morning or good evening, okay? Welcome everyone to today's discussion of the case study of DESTINY-Gastric studies. So first, I would like to introduce myself. I'm Sen Lu, Professor of Biostatistics at Duke University. And next, I'm going to introduce our next speakers, Dr. Jianqiang Cai, Associate Dean of China National Cancer Institute. He's going to give an opening remark. Let's welcome Dr. Cai.
Well, good morning, every expert. It is such a pleasure and honor to have this opportunity to communicate, exchange with so many experts in this field, taking this opportunity. And just with this academic platform, we can have such good seminars, both online seminars, to just exchange our opinions and the latest advances in the clinical trials of cancers. And also, on this platform, we can just have a discussion with each other to just progress the treatment of gastric cancer to a higher level. Especially we can learn something from Japan, which is a country with abundant experiences in treating gastric cancer. And to some extent, we will have very important help as well as support from these kinds of communications and learning. And also the curative or the response rate of treating gastric cancer in Japan is much higher in China. And that's just to give us more reason to learn from their advanced experiences. So from the opportunities of these webinars, we can just get more experiences to benefit the patients, not only in China, but also worldwide. And we would like to have a longer cooperation with different parts. And we also look forward to better outcomes of clinical trials. And for this seminar, well, we know that Professor Luo and Professor Zhao, they have done a lot of work in supporting this seminar. And here, I would like to say thank you again to Professor Luo, to Professor Zhao, and other organizers as well as the experts who support our seminar. And I hope that you will have a very good weekend to all the audiences and experts here in our seminar. Thank you.
Thank you, Dr. Cai, for your opening remark. Next, we are going to enter this roundtable discussion. So tonight's the format is that I will first introduce some key point of this study, and then I will welcome Dr. Shitara to talk more in details, and then specifically he'll answer some questions about the study design and how they designed the study in this particular way, and how they got all the important parameters and data when they designed the study. So, I think one question from the audience is that when they can ask questions, the answer is anytime. Please use your chat function to type in the questions, and I will moderate the questions. We will answer the question to the best of our knowledge. Without further ado, let's first start. So this is the study we are going to talk today. This is the gastric cancer from DESTINY-Gastric 01 study. So the study drug is the DXd. So this paper has been published in the New England Journal of Medicine in last year. So one thing that we need to pay attention is that this is a phase 2 study and then targeted for third-line therapy. And overall survival was used as a secondary endpoint. Now, the sample size was the 188. And then, so in January of 2021, this medication was approved by FDA as the second-line therapy, not the third-line therapy. And also this is the final approval. It's not accelerated approval. And this is very abnormal. So tonight we will going to discuss why this is the case. So I just want to spend a few seconds to talk about some highlight. So first of all, as I said before, so this is the third-line trial, but I received FDA's the second-line approval, and the sample size was the 188. Is usually smaller than most of the phase 3 clinical trial, and patient enrollment was pretty fast. It only took about 1.5 years from 66 sites, and the follow-up time from the last patient enrollment to data cutoff was 6 months. And it only took another 6 months to get the paper published in New England Journal of Medicine. So tonight we are delighted to get Dr. Shitara, the first author of the New England Journal paper, to share with us a lot of his experience and insight in this study. Let's welcome Dr. Shitara.
Thank you. So thank you for your very kind introduction, and it is my great honor to discuss about this trial in the DXd for gastric cancer. Next slide, please. Okay, so this is a timeline of this trial and approval of DXd. We started the phase 1 trial in Japan in 2015, and then we also started phase 2 trial in 2017. And during this trial, we also received a Sakigake designation in Japan. This is almost similar with a breakthrough designation in US FDA. And as introduced before, we achieved ASCO and the New England Journal presentation in 2020 in May. And in September, this DXd was approved in Japan as a third-line or later line for HER2-positive gastric cancer. In the US side, this drug received a breakthrough designation in May in 2020. And then early this year, this DXd was approved as a second-line or later line for HER2-positive gastric cancer. So next slide. So please let me remind you about this study. So first, this TDXd trastuzumab deruxtecan is a newer HER2 targeting antibody drug conjugate composed of a HER2 antibody, a cleavable linker, and a cytotoxic payload. And this DESTINY-Gastric 01 is a multi-cohort and a multi-institutional study conducted in Japan and South Korea. A primary cohort enrolled patients with a HER2 positive status by central assessment, and the patient should have received at least 2 prior lines of chemotherapy with fluoropyrimidine, platinum, as well as trastuzumab. And the primary endpoint of this study was objective response rate. And the key secondary endpoint was overall survival, which was statistically evaluated with a strict control over overall alpha error. So finally, 187 patients were randomized, and this is a 2-by-1 randomization. And overall, 76% of patients had IHC3+, and 80% of patients were from Japan, and 20% were from South Korea. And the median number of previous chemotherapy was 2. And actually, all patients received the fluoropyrimidine and platinum and trastuzumab, and the additional 86% of patients received the taxane, and 72% of patients received the ramucirumab, and the additional 1/3 of patients received the checkpoint inhibitors. So next slide. Thank you. So I have already received several questions. So let me answer each by one. So first question is about this is a randomized phase 2. So why not design this as a single study with response rate as a only primary endpoint? And actually, this design was similar to that of DESTINY-Breast01 study, and this breast cancer trial achieved accelerated approval. And this smaller study with a single arm design would lead to much smaller sample size with a possible accelerated approval. So my question was described here. So actually, initially we proposed single arm phase 2 study based on a promising phase 1 result, which showed response rate of 43% in 44 HER2-positive gastric cancer in later lines of treatment. However, the Japanese regulatory agency called the PMDA required randomization with control arm and the survival assessment. So we have several discussions, but finally, a company, Daiichi Sankyo, selected a randomized study. And we also discussed about a global study because not a few Japanese gastric trial have been conducted as a global study. But before this discussion, we have a very few patients into the phase 1 study from the US side. So safety and efficacy in non-Asian population was unclear. So our final decision is to conduct the Asian randomized phase 2 trial, and different phase 2, DESTINY-Gastric 02, was planned for non-Asian population. So next. So this is a result from our first in-human study. This includes 44 gastric cancer patients, and 39 patients were from Japan, and only 5 were from US. Median previous lines of chemo was 3, and because this is a first in-human study. But we observed a very good response. Response rate was 43%, and the response was a little bit higher in patients who received 6.4 milligram per kilogram compared with 5.4 milligram per kilogram. And actually, this 6.4 milligram per kilogram is a current standard dose for gastric cancer. In comparison, 5.4 was used for breast cancer. And on the right side, you can also see the PFS and the OS. PFS also favored 6.4 compared with 5.4. So this is a phase 1 trial, so overall survival is a little bit biased, but the actual survival in median was 12.8 months. And regarding the toxicity, notable toxicity is ILD, and actually we observed one patient with an ILD or pneumonitis. Next slide. So next question from this trial is dose of TXT. As mentioned before, we used 6.4 for gastric DESTINY-01 study based on this phase 1 trial. And this is different from breast cancer dose as 5.4. And my question was also described here. So first, we observed a little bit better response rate and the PFS with a higher dose in first-in-human study. We also evaluated the PK profile and compared it in gastric and breast cancer, and a little bit higher, lower PK profile was observed in gastric cancer compared with breast. So actually, this is the reason for this phenomenon is not clear. But previous trial like TDM1 as another antibody drug conjugate or pertuzumab as an anti-HER2 monoclonal antibody, this study also showed a little bit low PK profile in gastric compared with breast, and actually higher dose was used for gastric cancers. Next. So third question is one of the key questions. Sample size was calculated based on overall survival events in addition to response rate. By doing so, it would significantly prolong the trial duration. So what was the consideration to make overall survival as an important endpoint? And this may be the one of the key result for FDA to approve this. So my answer is, as mentioned before, statistical evaluation for overall survival was clearly required by regulatory authority. And we have some FDA communication after approval, and FDA person also say this overall survival result is one of the key findings to support approval. Next. So maybe I can start here.
Thank you. Yeah, I would like to give a little bit of background information. I know for some of the audience. So here I want to briefly discuss the single arm versus the control. When FDA will accept the single arm study. So here I highlighted the important point when there is no available therapy, and where the major tumor regression can be attributed to the tested drug. And in these two cases, so FDA may accept ORR as the primary endpoint, and then the study can be a single arm. Okay. However, in this case, since this is a single arm, you could present the survival information such as overall survival and DFS and PFS and so on. But the survival information cannot be interpreted. Okay. So also about the FDA's accelerated approval, here are some conditions here. So first, it has to be a serious condition, right, to address the unmet medical need. And also you have to demonstrate the meaningful advantage over the available therapy. And also, if you choose the ORR as the endpoint, so you can use the ORR endpoint as to reasonably predict the clinical benefit. And about the sample size, I think this is a good time to have a discussion with the FDA or other regulatory agency. Now, specifically, FDA had the guideline. Within this guideline, there is a chapter about accelerated approval. So about this accelerated approval, I want to use the DESTINY breast cancer study as the example. So they received the accelerated approval in 2019. So here is the accelerated approval document from FDA. So here in this breast study, so ORR is the 60%. And then, as we mentioned before, because this is a single-arm study, and you can see from here, the FDA's guidelines say that the survival-related endpoints, such as the PFS and time to response or overall survival, cannot be interpreted. Okay, and then also as a requirement for the accelerated approval, the drug company has to conduct a phase 3 study to support the positive result. Okay, so this is one of the preconditions. So now, and that's the Another interesting thing from this study is that we noticed that in the protocol, so the old design has the 3 endpoint with the OR as the primary endpoint, and then you have a PFS and then OS, right? And then tested in this order. But in the new design, they remove a PFS. So we are going to spend a little bit of time to discuss why they changed the old design to the new design by removing PFS. So yeah, Dr. Shihara.
Yeah, thank you. So I myself and Daiichi Sankyo think PFS is one of the important endpoint for cancer patient to delay the progression, but this is not the case for regulatory authority. So again, PMDA strictly require overall survival result, and there is a several background. First, in gastric cancer field, we have no good data to support PFS as a good surrogate endpoint for overall survival. This may be a little bit different from other cancer types. So I'm the member of gastric as a meta-analysis group to do IPD, so patient data-based meta-analysis, but here we don't have a very good correlation in previous trial. And the other point is described here with gatekeeping strategy with response rate PFS and the OS sequence, we may miss a chance to assess overall survival if PFS was marginally negative. So finally, these two major reasons for us to remove PFS from key secondary endpoint, but PFS was still analyzed as one of the secondary endpoint to show, to calculate the hazard ratio or to see difference in median value. Thank you.
Thank you. Yeah, so about the gatekeeping, here is the FDA's document about gatekeeping from the multiple endpoint in clinical trial. So basically, you have to test the hypothesis in a particular order. In this case, let's look at the new design. We have to test the ORR first, and then if it's significant, and then you can test the OS. But on the other hand, if ORR's the hypothesis testing is not significant, you cannot test the hypothesis for OS. So another interesting point is that about the sample size calculation. So here, based on the results in the protocol, we can calculate the sample size when the power is 80% and versus the power under sample size of 120 in treatment group and 60 in the control group. We can see that the ORR has the highest power under this sample size, and then the PFS has and OS has the similar power. Okay, so in this document from FDA about how to choose the endpoint, so there are 2 principles here. One is that the endpoint can be ordered by clinical importance, because in this gatekeeping study, it's quite important to decide which one go first, which one go next, right? So order them by clinical importance is the one principle. Another principle is that you can order them by likelihood of success or demonstrating an effect. So in the previous case, the ORR has the higher power, so it's pretty reasonable to put ORR first and then OS as next. So I think this is a good point we can discuss and open up the panel. So I think, Minghua, you want to bring up the slides and then so that we can introduce all the panelists today? Yes, today we are also very delighted to have Dr. Hong Zhao from China National Cancer Centers and Dr. Ying Yuan, Professor of Biostatistics from MD Anderson Cancer Center, and Dr. Li Tang from Shenzhou Children's Hospital. She's also a statistician, and Dr. Ling Zhao from Peking Union Medical College Hospital. We can go back to the to the question here. So I think one question here is that the OS data not available in 2017 July, right, when the protocol, the first version of protocol was developed. And then was that the reason that PFS was and OS was both selected as the endpoint?
Yep. So this is a little bit complicated. So going back to the first-in-human phase 1 study, so this is a phase 1 trial. So to collect the survival data and assessment for overall survival is not usually included in the protocol in general. So we first analyze response rate and the PFS. So this is a more quick efficacy endpoint. But during this discussion, PMDA and the regulatory authority require overall survival. So we also try to collect the survival data in first-in-human study. So actually, the protocol in first-in-human study was revised to include overall survival data to collect. So for first question, OS data was actually not available. So maybe we can discuss this later. So actual estimation for overall survival is a little bit different from our expectation, and the actual result a little bit longer. Overall survival was observed in both experimental arm and control arm. So during
go ahead.
Revision and protocol, protocol revision, we have actual data from first-in-human study.
Yeah, thank you. So here is to show the time of a different version of protocol. The original version was developed in July of 2017, and the data cutoff day was August of 10 of 2018. So here to show the cutoff day. I want to skip all this. Here I think this is a very important point we can really discuss in the panel. So here, why we started at the third line and not a second line in phase 2, right? So here I sum up all the results. So maybe we can start from Dr. Zhao. So maybe you can give us some insight about this. Why would you think they start from a third line therapy instead of second line, given the promising phase 1 result?
I think the most important fact is that we can see the OR is very similar between the second line and the third line. So, and OR is the first endpoint of this study. So, I think maybe it's a most important factor that the investigator choose third line as the targeted lines of therapy.
Any other insight, Dr. Yingyuan?
Yeah, I think Dr. Shitara probably holds the answer. So he has the answer. So for us, it's just a quiz. We just guess. Okay. So and Shitara can say who have right answer at the end. My guess is typically when you're treating those patients, you always start from late line, because that's you have a biggest chance to be success, and also when you start from the later line, and especially if you do the single arm, and if I think it's have a less ethical issue because those patients are essentially out of choice. And also you can see in this trial, they do 2-to-1 randomization. And I think that's also part of the reason they do 2-to-1, because for those patients, they really out of choice for good treatment. So that's my guess. And
the other insight?
Professor Lo, we have medical oncologists here, and Professor Chunxia Du, she wants to raise a question or give some discussion.
I have the similar guess like the last professor. I think the third line therapy is easy. Choose the third line therapy is easy because if we choose the second line in phase 2, we have to choose maybe paclitaxel or irinotecan as controlled therapy. So it was not so easy to get a positive result. Thank you.
Dr. Shitara, there are a few questions in the chat box. Maybe you can give us some insight here. So first question is about how many sites in US for phase 1? What is the efficacy for the 5 US patients in phase 1? Do you have this kind of information?
I forget the exact number of sites, but maybe 1 or 2. MSK and MD Anderson or something. Yeah, very few sites.
Yeah. Do you have the efficacy data?
So I'm not sure about that. But, the 44 gastric cancer patients include the 5 patients in the US. And I heard that there's 2 or 3 patients with response, but I'm not sure about that. So can I make a comment for the table and previous topic?
Yes, please.
About
please.
the second line versus third line. So let me introduce the table here. So first table, phase 1 result is the actual result from TDXD trial, 43% response rate and the overall survival around 1 year. And this third line therapy is based on historical data with other available therapy like irinotecan or taxane's agent. Usually response rate was around 10%, and the overall survival was very, very short. And in second line, we have a global standard based on Rainbow study with paclitaxel and ramucirumab. Ramucirumab is one of the VEGF inhibitor, and in combination with paclitaxel showed the survival benefit compared with paclitaxel alone, and the response rate was around 30%. And this is a very good drug. So as mentioned here before, third line is more easy for us to have a positive result. And in other words, third line is a more unmet need population. We need a better drug. And actually this phase 1 result was based on third line or later line. And other final important point is a difference from irinotecan. So looking at the detail of TDXD, this is a combination with anti-HER2 and the topoisomerase inhibitors. And irinotecan as a key drug at third line is also topoisomerase inhibitors. So some regulatory person ask exact difference with TDXD versus irinotecan as a same cytotoxic drug. So that's why these are main reason for us to conduct the third line and later line trial.
Thank you. Yeah, so here I listed one disadvantage of the old design when we have PFS, right? So here, because if you put PFS there, we might need to wait for all the PFS event to reach the target number before we can perform the overall survival, the first interim analysis. So it may take more time as compared to removing a PFS. Right. So here I give some information about the first interim analysis results. So the OS, about OS. So it take about 24 weeks from May 2019 to November 2019. So here is the result. So I think the key question here is that, right, so when they performed the first interim analysis, the PFS event was the 109, right? So not their target event of 128. If they did not remove PFS as one of the endpoint, if they still use the old design with the PFS as one of the endpoint, right? So they will have to wait from 109 to 128, right? So that I'm not sure how long it might take to reach that target number. Dr. Shitara, would you have some insight here?
So yeah, this is a very important point. And this PFS was based on central assessment and not based on investigator judgment. And actually, almost all patients with a control arm discontinue treatment at this timing, but some cases were censored, as you can see from this Kaplan-Meier curve. And this is also same for experimental arm. So patients discontinue treatment because of progressive disease by investigator judgment, but this was sometimes judged as a non-disease progression by central assessment. So finally, that is why the actual event is a little bit smaller than that of expected event for PFS. And we already presented updated data for PFS and OS in ASCO this year, but the final PFS events number was only 118. So this is still lower than that of 128. So I think this is one of the issue in PFS assessment, especially in gastric cancers.
Yeah, so I'm going to skip this. This is the some detail about how the interim analysis will perform. This is the final result for the OS in the first interim analysis. So one thing to notice is that the event number at this point is the 101, which is about 76% of the target 133 event. Because this is interim analysis, that they have to calculate the boundary and then to decide what portion of the alpha is going to be used in this interim analysis. In the protocol here is that the boundary is the 0.02. I think that Dr. Shitara, you can tell us more about this result.
Mm-hmm. So first, primary endpoint, the response rate was clearly improved with TDXD, with 51% in TDXD arm compared with 14% in control arm. So I want to mention that this includes unconfirmed response rate also. Confirmed response rate was 42% in TDXD and 43% in TDXD and 12% in control arm. So in RECIST guideline, there is some description that in randomized study, we can also use the unconfirmed response rate as a primary endpoint. And in single arm study, we should use confirmed response rate as a primary endpoint. But anyway, both response rate are clearly improved with TXD. And we planned overall survival interim analysis at this point, and hazard ratio of 0.59. And as mentioned previously, the boundary for overall survival p-value was decided based on the event number as a 0.002, and this was, and actual p-value was less than 0.02. So this at this time we can say that overall survival was also improved with TDXD. And this, as mentioned before, this was based on 101 number for OS events. And we also reported the final overall survival at this ASCO, and this include 133 event, and the hazard ratio for overall survival was 0.60. So very similar, 0.59 for interim analysis and 0.60 in final analysis. Thank you. Oh, sorry. So I just want to say that the efficacy endpoint in phase 1 and phase 2 was very identical. Response rate 43% in both phase 1 and phase 2 in terms of confirmed response rate. And the median PFS 5.6, median overall survival around 1 year. So at this time, I still wonder whether we need a randomized study or not. But anyway, this randomized study is give us a more confident result to show survival benefit and response rate. But anyway, the efficacy was completely reproduced in this phase 2 from phase 1 trial. Next.
Yeah. Can I ask a quick question? So,
yes,
so
please.
this is so this is amazing. The results are so consistent. So in the phase 1 and phase 2, do you use the same eligibility criterion or any modification on that?
Thank you. So actually almost same, but one important point is how to assessment. In phase 1, we can enroll patients with how to assessment at the local site. But in phase 2 study, a central assessment was performed using the most recently available tumor sample. This is one of the major difference.
Thank you.
Thank you. Yeah. So here we can calculate the sample size based on the protocol results. So as you said, the estimated effect size was for ORR was 40%, was 15%, but the final result was very close to the estimate. And then if you look at the PFS, it's the same thing, 5 months versus 3 months. And the final result is the 5.6 months versus 3.5 months. Very close. OS, the result is a bit interesting because the estimate was the 9 months versus 5.5 months with the HR of 0.61. The final result was close in terms of the HR 0.59, and the OS was a little bit different. So here we sum up the results from phase 1, phase 2 results, and also the estimate. Overall, you can see the results are very consistent. So here I'm going to skip the sample size calculation. So if you are interested, we can send you the slide. You can reproduce. So here we want to discuss about the sample size estimate. So I think most of the audience are very interested in the first question. So because when they design the later phase studies, though I think one key important question in terms of calculating sample size is how to specify the effect size. Most likely they have to estimate the effect size from the early data or historical data, right? So let's open up the panel and see any insight here, how we do this. Maybe we have Dr. Ling Zhao. So I'm not sure if Dr. Ling Zhao is also in the audience. Can you speak up?
Hello, Dr. Luo, I'm here.
Yes. Okay, yeah. Okay. Any insight about any of these questions?
Yeah, from my experience, normally I design a clinical trial. The first question is to estimate if the sample size. Yeah, it's very important. And normally we'll do many to review the data has already published, and after that we can know what's the control control arm the response rate, and also estimate the experiment arm what expected. And after that, we know the difference between the two arms. After that, we can estimate the sample size. So normally we did that, but it is still many questions. First, normally we can maybe we estimate a very large sample size, but in reality, in real world, maybe it's not. We couldn't do so big sample clinical trial. So after that, it's very difficult to decide which way we should choose. The first way is we add the difference. But sometimes it is very risk. I think we have to. It's very dangerous because maybe the experiment arm is not so perfect. So maybe you will the clinical trial will fail. Another way is we cut the sample size, but if you cut the sample size, it's also very dangerous. But if we want to do the big sample size experimental, you have to have enough sources, I think. Yeah. So this is my question. I don't know how to do the perfect estimate the sample size. Thank you.
Now, Dr. Li Tang, so you have designed many clinical trials in St. Jude Children's Hospital, right? In your hospital, you have the largest children cancer cases. Now, you design many early trial and also the late phase trial. So about this, the effect size, what's your experience about this?
Thanks for the question. Can you hear me well?
Yes.
Is my voice low? Okay.
Yes.
I agree with Dr. Lin Zhao. I think this is actually a very tricky part in designing a trial. In this particular trial that we are seeing today, actually the phase 2 results confirmed were very close to what were observed in phase 1, which is amazing. But in my reality, I don't think that always holds. So estimating the effect size of the later phase trial from early phase data, or estimating the effect size for the early phase based on the historical data, both are not. I don't think there is a straightforward answer to that. What I personally, maybe that's not the best practice. What I personally usually do, for example, if we want to estimate the effect size based on the historical data, when the trial is still pretty early, we don't have a lot of trial data from this particular population, particular indication, particular medication. Usually there is data variability. There is variability, study to study variability in historical data. There's a range. I personally tend to go more conservative when there is a range. Try to use the more conservative estimate, the smaller size. And this leads exactly to the issue Dr. Lin Zhao just mentioned. This may lead to a large sample size sometimes. But on the other hand, if we aim for the success of the later phase trial, I still feel that is a safer choice. And a similar principle can apply to the early phase data. There might be multiple similar trials conducted, early phase trials conducted. There might be a range for the effect size estimate, I personally tend to go to the low end, which may inflate the sample size of the late phase trial a little bit. That's what my practice. But I agree, this is a very tricky question.
Thank you for the answer. Now, Dr. Ying Yuan, so another question I have for you is that about the optimum dose, right? You know that in phase 1 study, so you want to find you most likely find the MTD, but MTD may not necessarily be the optimum dose, right? But also optimum dose is critically important for the success of phase 2 and phase 3. So can you share with us any insight about this point?
Yes. So early phase it's always a challenge because of very heterogeneous population and also limited sample size, right? And I think, for example, in this trial, I think in the phase 1, I guess they use all comer, and then they do a co-expansion in different indication. So I think for this one, first actually, originally I thought I'm going to follow the previous question. So I think the first thing is you should find a good PI like Dr. Shitara. Okay, so yeah, actually congratulate for such a beautiful work. It's perfectly consistent. Between early phase, later phase. So I think the first thing is the PI should realize there is no free lunch. So if you want to optimize dose, get a reliable estimate, you must treat enough number of patients, get enough data. Statistician cannot really, it's not a magician. So what we can do is we get enough data. We give our best estimate to help clinician to design the trial in the best way. So that I always say, for early phase trial, you must more patient and be patient. Should get enough data and good data to, like in this trial, and you can see they finished the phase 1. They don't really rush into, pick one dose. Let's do quick co-expansion and finish the trial. So in this trial, I think they choose 2 doses, right? Do co-expansion, doing more careful work to study the PK to PD and identify, which dose is really optimal in terms of a risk-benefit trade-off. So I think once we do that, and we will have better estimate and help us to plan our phase 2, a large phase 2B or phase 3 study that will improve success. And of course, I think in practice, there is always a lot of uncertainty in terms of those effect size. Especially, I think in this trial, as Dr. Shitara said, the patient population almost similar. But in many cases, phase 1, we put a more homogeneous patient. In phase 2, we have a more specific subgroup. And in that case, it's more challenging to estimate effect size. And I think in that cases, we can do best we can. And also for later phase, we can do some adaptive design. So in the case, for example, your early estimate is not as you anticipated. You still have a chance to rescue your trial, do your best job. So that's my comment.
Thank you. So my next question is about how to estimate the enrollment time and follow-up time. So this question, let me invite Dr. Jianqiang Chai's group. Any doctor from your groups can answer this question. So when you conduct a say, multi-center study, how do you normally estimate the enrollment time and follow-up time?
It's kind of easy to estimate the follow-up time since this is a second-line therapy, and for gastric cancer, we also know that we all know that the survival time for patients in the later line therapy, the gastric cancer patient, the third-line therapy, the survival for this kind of patient is usually less than 6 months. So it's kind of easy to estimated follow-up time. And for enrollment time, I think it's very hard for a clinical clinician to estimate it. We need the support from statistic people. Thank you.
Thank you. Okay, let's move on. So Dr. Shitara, so can you tell us about the design questions?
Yeah. So, yeah. Before that, could you go back to the previous slide?
This?
Yeah. So I just want to say that this is a very difficult question, but this should be based on cancer type and based on other available therapies. So actually, I enrolled more than 20 patients for phase 1 trial and see the response in exact case. And this clear tumor response should not be achieved with other available therapy for gastric cancer in same treatment line. So I completely agree with the previous comment that ideally we need a more sample size to achieve the good activity or to estimate the effect size. But from our experience with this drug in this trial setting, we are confident to move forward. And regarding the final question about the FDA approval, this may be the question for FDA, but FDA think confirmed response may be more important as a clinical benefit. And this is one of the unique phenomenon with this drug. Around 10% show the early treatment response followed by early disease progression. At this time, we are not sure about the mechanism for that. Maybe based on intertumoral heterogeneity or other resistant mechanism. But anyway, FDA think duration of response may be also important. Thank you. So next slide.
Thank you.
Yeah. So the question I received next is about the difference of actual number of patients in response assessment compared with our planned sample size. And this was based on the 12 patients who are excluded from central assessment because of patients with a non-measurable region. So patient could be enrolled by each investigator if investigator judged patient with a measurable region by RECIST criteria, but this should be assessed by central radiologist for primary endpoint, and actually 12 patients were excluded from this analysis. And I want to mention also difference for the confirmed response rate in final assessment and primary analysis published in New England Journal. In New England Journal, we said that confirmed response rate was 42%, but in final assessment, it was 41%. So because this was based on one patient who finally judged as a stable disease. At the initial assessment, this patient was judged as a PR, but at the final assessment, radiologist reviewed the CT scan again, and this patient was considered as a stable disease. But anyway, there is a change in response rate in 1%. Next slide. Yep. So this question is about enrollment time and determination for follow-up time. And our answer are described here. 133 deaths was considered as needed to observe the 80% power to detect the hazard ratio of 0.61. And actually, our estimation for median survival was 5.5 for control arm and 9 months for TDXT arm. and this is actually shorter than that of actual result in phase 2. But as mentioned before, usually we enroll selected patients for phase 1 trial, especially first in human study, and we expand the patient cohort even with the same patient criteria. So that's why we estimate a little bit shorter median survival, but actually the result are more better. And the sample size of 180 subjects also provide 81% power to detect the overall survival, assuming 1 month enrollment, 10 months enrollment period, and 1 month follow-up. I believe 1 year follow-up. I believe 1 year follow-up is usually enough for gastric cancer population at third line and later line to observe enough events. Next. So this is also a little bit complicated question, but yeah. So first question is about overall survival result. As mentioned before, actual overall survival was longer than that of expected result. Response rate and PFS was very identical in our estimation and exact result. And I answered here about longer overall survival is especially based on newer other therapy like nivolumab or other checkpoint inhibitors. And this treatment was used in 31% in T-DXd arm and 45% in control arm. But even with a little bit smaller population in T-DXd arm to receive the checkpoint inhibitor, we can observe the better overall survival. And again, despite longer median overall survival in control arm, we can observe the clear survival benefit in T-DXd arm. And the next question is about 2-to-1 fashion. This was also mentioned by other panelists here. So patient, based on our very promising phase 1 result, patient want to receive the T-DXd, and actually not a few patients were referred from other hospital to our institution to enroll into the trial, and the patient hope to receive T-DXd. So, but this is a randomized study, so finally we decide to 2-to-1 fashion. Thank you. Next question is about FDA comment. So I recently joined this workshop with the FDA. And we discussed about a process to get approval of T-DXd, and FDA staff suggested that this approval is based on similar patient population defined by biomarker and a similar standard treatment and a trial result itself. And actually, a reviewer from New England Journal also say similar comment that usually it is unrealistic to do another randomized study in non-Asian population because we clearly observed a very good result. Thank you. So maybe next topic is about approval difference for indication. In Japan, regulatory authority decided this treatment as a third line for later line because of our DESTINY-Gastric 01 is based on this setting, third line later line population. But US approval also allowed to use this for second line or later line, and they also recommend biopsy to confirm the HER2 status at the second line. And our trial do not do fresh biopsy. We usually use the archival tumor tissue for central assessment for HER2, but FDA had this recommendation. So that's why we did additional biomarker or subgroup analysis for this trial. Next slide. Yeah.
Can I ask you a question before you move to the next one?
Yeah, sure.
Yes. So since FDA approved the second line, do you contact with PMDA about request also expand the indication, the label to second line, and how the PMDA responded? I just curious.
Thank you. So honestly, we have not done such communication, and I expect this is usually not possible for PMDA because PMDA is a little bit strict, and usually they decide indication based on trial itself. So honestly, we did not expect this FDA decision to allow second line because our trial is a third line. So FDA decision is a little bit tricky for us.
All right, thank you.
So this is a biomarker subgroup analysis. As mentioned before, we usually use the archival tumor tissue to test HER2, but some patients used archival tumor tissue before first line, and this is in 70% of patients. And 30% of patients, for 30% of patients, use tumor tissue after trastuzumab therapy to confirm the HER2. And why this subgroup analysis is important? So some research suggested loss of HER2 positivity after trastuzumab treatment, especially on disease progression. So we conducted this subgroup analysis by looking at the response rate by this subgroup. We can observe the clearly higher response rate even in patients who have archival tumor tissue before trastuzumab treatment. And a little bit higher response was observed, especially confirmed HER2 status even after trastuzumab, but 49% versus 57%. But clearly, this response was still higher in T-DXd arm. Next slide. And looking at the overall survival, the hazard ratio for treatment effect is a little bit larger in patients, especially confirmation of HER2 after trastuzumab treatment. I believe this is maybe one of the reasons for FDA to recommend confirmation of HER2 status after trastuzumab. But we can still observe the hazard ratio of 0.76 in patients with a HER2 positivity using archival tumor tissue before trastuzumab. And also looking at the survival result in T-DXd arm in both subgroups, very identical. 12.1 months is for tumor tissue before trastuzumab and 12.5 months is in patients with tumor tissue after trastuzumab. And Kaplan-Meier curve also showed a very similar result. So I personally think if we use this drug at the third line or later line, fresh biopsy may not be necessary, but the FDA decide based on this result to recommend fresh biopsy, especially to use this as a second line. Next. So this is out of a discussion point, but anyway, toxicity is usually important for us to decide the use of treatment. And one of the notable toxicity of T-DXd is pneumonitis or ILD. And around 10% of patients had a pneumonitis, and it was increased to 16 patients at a longer follow-up. But most of patients had a little mild grade 1 or grade 2, and very few grade 3 or higher events. Next. Yeah. So the activity. So can I continue without any additional discussion?
Yes, please.
Okay. So this DESTINY-Gastric 01 was based on Asian study in Korea and Japan. So the activity of this agent was further confirmed in additional DESTINY-02 study. This is a single-arm study in non-Asian countries and actually second-line trial with a confirmation of a HER2 status in fresh biopsies. And a little bit different observed here is 34% in gastric and 66% from GEJ gastroesophageal junction tumor. This was different from DESTINY-Gastric 01 in Asia. We observed more gastric than GEJ. So this is a usual difference in gastric cancer in Asia and non-Asia. And the confirmed response rate was 38% in this non-Asian trial and 43% in initial presentation of DESTINY-Gastric 01 and 42% in updated analysis. So a little bit lower in this non-Asian population. Median PFS was almost similar. So I personally think the different efficacy was confirmed in this non-Asian study, but some investigators may think this is a little bit lower response rate compared with the Asian trial. Next.
Can I ask a question before you move on?
Sure.
Yeah, so if I'm right, in previous slides, it seems Western, non-Asia people have better tolerance to this drug. Is that true? Seems to have a much lower adverse event.
Yeah, this is a very important comment. So could you go back to the next previous slide? Yeah, this is a summary of safety in DESTINY-Gastric 01 and 02 in non-Asia. So one of the very unique difference is a difference in neutrophil count decrease. 63% for overall grade in DESTINY-Gastric 01 and 51% for grade 3 or higher. But this was much lower in DESTINY-Gastric 02 study. So GI toxicity is almost similar, but a little bit lower in 02 study. So maybe one question here is a dose or PK profile between Asia and non-Asia. And actually, I don't have enough information here. And maybe one idea is to try higher dose for non-Asian population, but I'm sorry, I'm not sure at this time.
So is that common in gastric cancer? Like for existing chemo, Western people tend to have better tolerance.
Yeah, thank you. This is also very important comment. So yes, right. So usually a Japanese or Asian population have a better response rate or better outcome, but at the same time, higher incidence of toxicities for bone marrow suppression. Yeah.
Yeah, maybe increase those a little bit. 02 going to have a similar response as 01 because they are different by 5%, and also you have lower adverse event. Maybe a little bit higher dose, you can achieve the same efficacy. So
maybe.
Yeah, thank you.
Thank you. And actually now global phase 3. Oh, sorry, next slide. Now our phase 3 trial is ongoing, DESTINY-Gastric 04, to compare T-DXd and the second line trial. And the major difference from Gastric 01 and 04 is a global versus Asian trial and the line of treatment and the central HER2 confirmation. And so maybe the important question is why we conduct this trial after previous 01 study. So this is yes, this is a question. So first, FDA decision to allow second line is a little bit unexpected or tricky for us. And that's why after FDA decision, the US is excluded from Gastric 04 study. And importantly, phase 3 is still required in not a few countries to get approval. So this should be the most important reason to conduct this Gastric 04 study. And in Japan, as mentioned before, the indication of T-DXd is at third line, later line. So therefore, study is necessary to expand this indication to second line.
this is the subgroup analysis result. So here we can see that from the subgroup analysis, these three are the stratifying factors. So in the Gastric-01 study, so we see that in Japan versus Korea, so the results are quite similar. In AOCRAS scores for 0 or 1, the results are also relatively similar. However, in the HER2 expression, the cohort with the IHC 3+ versus the IHC 2+, so the results are quite different. So from statistical standpoint, now after seeing this result, we can say, well, it looks like we do not need to have the region or AOCRAS score as the stratifying factor. However, stratifying by this, the HER2 status seems to be quite important here. Dr. Ying Yuan, do you agree?
Yeah, but I think this is just when we saw the data, we post hoc can make that decision. But a priori they don't really know if there is a region difference. Or I think ECOG typically it's a pretty standard important prognostic factor. I don't know. If Dr. Shitara has any additional comments.
Yeah. So clearly, so this is a exploratory analysis, but still HER2 status should be important to predict efficacy. So we also evaluate the activity of this agent in HER2 low population, including IHC2+ with fission negative or IHC1+. So these overall results clearly suggest that the IHC status is a very good predictor to efficacy. But still, we observed 29% in IHC2+ in each positive population. So I believe this is still clinical meaningful efficacy in this setting.
Yeah, also, I'd like to ask Dr. Hong Zhao or Dr. Ling Zhao. So any of you have any comments about this point, how to do the stratifying factor? This is more like a clinical question.
Yeah, Professor Liu, I have a question. Yeah, I also very interested in the primary cohort about according to IHC, and I think if the IHC is 3+, the DTXT is more effect. But for the IHC 2+, I think the control arm, the response rate is too high, because the sample size is very small and the response rate of chemo is 25%. I think it is much higher than we know. It's much higher. If this is also 11 or 12%, I think it is also still the meaningful difference between these two arms. So I don't know what's opinion of Shitara. What do you think of this difference? I think this 25% is too high.
I completely agree with you. This is based on very small patient population, and 25 is a little bit higher than expected. And we don't have an exact explanation for this phenomenon. We cannot expect a better cytotoxic drug activity in HER2 positive population. So compared with the IHC3+, so there should be no large difference. There should be chance.
I want to know in Japan and in United States, the approval is also both for IHC3+ and IHC2+, yes or not?
Yes, approval include IHC2+ and each positive population also.
Okay, thank you.
So
Dr. Shitara,
question about the stratification factor in GC01. HER2 status region and equal PS was used, and the GC04 also include HER2 status, geography, and interestingly, time to progression on first line was also included here, because this is a second line trial, and some previous research suggested the prognostic value of this PFS or time to progression on first line to predict the outcome after second line therapy.
Yes, here, we'd like to open up the panel to discuss any of these questions. So any comment for any of the questions? Anybody?
So I think, if no one, I can talk a little bit. I think for the first one, we have already discussed that quite a bit, right? It's because O2 and O1 are in different population, Asia versus non-Asia. Due to ethnic difference, a lot of time, I think they have a different PK profile. So, as Dr. Shitara, just said, I think in gastric cancer, it's quite a common, patient, Asia and non-Asia respond differently. Asia tend to be have better efficacy, but also higher adverse event. I think for the last one, just my guess, probably Dr. Shihara can give a better answer. I think it's kind of standard. People use the region as a stratification factor. Especially, I think lifestyle and in China, it's very different from China. So I think it's definitely important to stratify and on that factor. How to estimate O4 effect size? O4. So O4 is second line, is that right? Because there are so many O1, O2, O3, O4.
Yeah,
O4
that's right, second line. Yeah.
It's the second line randomized phase 3 in Asia patient. Is that right? If that.
Yes.
I don't know. I think for that one, of course, if you have some data, definitely you can base on that. If not, I think you can just base on historical data or literature. I think for that answer, for that question, I think maybe Dr. Shitara is the best one to respond. What they have, what's their consideration when design 04.
Yep. So there is a statistical hypothesis for also. Maybe we have it in next slide, right? Yeah. So the primary endpoint is overall survival for this 04 phase 3 trial, and estimate overall survival was 15.4 versus 10.8 months. So we don't have any good rationale for that, but this may be more based on clinically meaningful difference in second line. And when we use T-DXd at the second line, we can also use paclitaxel and ramucirumab as a third line treatment. So I believe we can have a chance to achieve this median survival in T-DXd. One concern or comment here is that in Japan, we can also use T-DXd after paclitaxel and ramucirumab, almost like a crossover treatment. So this is one possibility to dilute the overall survival effect. But this is a global study to enroll more patients in other countries where T-DXd has not yet been approved. And the other question here is how was the interim analysis? And here our answer is that we expect a 70% planned over survival event at interim analysis. Thank you.
Yeah, I think that's a very interesting point Dr. Shitara mentioned. Is I think if the Japanese patient can have access to TDXD, in the control arm, that's gonna be have a pretty good big impact when you compare final OS, right? Because it's such effective drug. So I'm wondering, can you put those patients as a sensor? Because a lot of time when patients receive salvage treatment, you can instead of you continue follow their OS, because especially in this case, it's the same drug and it's such effective drug. And I don't know if you guys think about those patients, you can treat it as censored after they, in the standard care arm, if they receive the savage treatment, they use TDXD, treated censored, rather than continue follow them. You can still follow them, but when you analyze, that will help you, to avoid that dilution of treatment effect.
Yeah, thank you for your suggestion. And actually, I'm not a statistician, but I know that there is a trial for to include such kind of analysis. And at this time, our expectation is that still small number of population receive TDXD at the third line, because again, this is a global study, and maybe we decide to cap the enrollment from Japan to make the impact of this crossover small. But anyway, we should take into account your suggestion in future analysis. Thank you.
Any comments about this slide, Dr. Shitara?
Oh, yes. So this is just a summary of ongoing trial for gastric cancers. So HER2 is still very relevant target. And as mentioned here, DESTINY-Gastric 04 is a second line trial, but now additional studies are also going to combine this TDXD and other agents. And then maybe in future, we will also consider first line trial with TDXD in combination with other agent. And I know in China, this RC48 is also very important neuro HER2 ADC, which shows a promising activity in second line, later line. And I heard that this drug is approved in China, and this was also now evaluated in trial. Thank you. Oh, actually, this is a trial I mentioned to evaluate combination with TDXD and other agent, including other cytotoxic agent or checkpoint inhibitors. And next.
The Keynote A11 first line treatment just got accelerated approval with the ORR of 74%. Yeah. Okay. Now, Dr. Shitara, so I think due to the time, now I think one very important question a lot of audience ask is that they look at your CV. They were amazed by all the papers you publish in New England Journal, JAMA, and Lancet, and many other high-impact medical journals. So can you maybe use the last few minutes to share with us your experience and your insight about how to publish in those high-impact journals?
Yeah, thank you. So, yeah, this is not only for my work, but this is usually based on effort for many investigators and companies to develop new agent or new treatment for, especially for gastric cancer. My major is for gastric cancer. Still gastric cancer is a very difficult disease to treat, but these efforts build up the knowledge and the new treatment. And I can briefly share some experience in this New England Journal publication. We have a top line result in January 2020, And before, so this is 6 months before our publication in New England Journal and ASCO. So after we have a top line result, we send pre-submission inquiry for New England Journal in February, and they are very positive about that, and they recommend us to submit a full paper as official submission. And as a submission, we also mentioned that we try to have a simultaneous presentation in ASCO and the New England Journal, hopefully. And they are also supportive for that. So our paper was reviewed in very quickly as a fast track review. So we received a first review in 5 days after our initial submission. So this is a very short compared with other experience for New England Journal submission. And the first decision is to ask us for revision, but all the reviewers' comments are very positive and reasonable. And as mentioned before, some reviewers say that we cannot do a same trial in non-Asian population. I worry about for that because this is an Asian trial, mainly in Japan. So New England Journal say, oh, this is a Japanese trial, Asian trial, so we should have a non-Asian result, so we should reject. But this was not happened. So they are very positive. So I believe we can have such achievement in future in other Asian trials, for example, in Japan and China collaboration or Chinese only trial. So always we have a chance in gastric cancer field. I think this is one example. Thank you.
Thank you very much. Due to the time, so let me just quickly talk about, tomorrow we are going to have a very interesting discussion about who will be the next king of the first-line treatment of gastric cancer. So we will invite Dr. Yo-Wen He from Duke University to talk about the mechanism of immunotherapy. And then he will make some interesting prediction who will win in this game. I think we can end here. So thank you very much, Dr. Shitara, and for sharing with us your experience, your insight. I really appreciate everything you have said. And also I want to thank all the panelists for your insightful comments and the audience for your questions. Thank you very much.
All right. Thank you.
Thank you.
Thank you. Bye.
Thank you.
Maybe the last minute to talk about this. We would like to bring your attention to this, the eChinaHealth news. And also next page. So they also have the conference. This is the page of all the speakers. Please pay attention to the time if you are interested in this conference. And next. And also in January of next year, so we are going to have a discussion with Dr. Sarah Hovich about the same medications, breast cancer trial. So please attend this if you are interested in this. And next. Yes, thank you very much, and thank all the sponsors and all the audience. Thank you very much. Thank you, Dr. Shitara. Hopefully we'll see you soon and for more discussion. Thank you.
Yeah, thank you.
Bye, everyone.
Arigato.
ありがとう。